welcome all to today's session of the Virtual Journal Club of the Journal of Physiology. My name is Thibaux Van der Stede, and I'm a PhD student at Ghent University in Belgium under the supervision of Dr. Wim de Rave, and also at Copenhagen University under supervision of Ylva Helsten and Lasse Gliman. I will be your host for today, and I'm very happy to introduce the paper that we're going to talk about today. It was a paper that was recently published in the Journal of Physiology. But I'll start with introducing our panel for today. These are the two lead authors of the paper, Dr. Peter Tickle, who is a postdoctoral researcher at the University of Leeds in the UK, and Dr. Paul Hendrickse, postdoctoral researchers at Manchester Metropolitan University, also in the UK. And we also have one of the editors of the paper with us today from the journal who worked on this paper, Dr. Caroline A Rickards from the University of North Texas Health Science Center based in the USA. So thank you very much, all three, for being here today. For those of you that might be new to the format of the Virtual Journal Club, I'll briefly introduce you to how today's session will run. In a moment, I will hand over the words to the lead authors who will present some of the key findings and the significance for the field. After that, Dr. Rickards will discuss some of the key elements that made this particular paper suitable to be published in the Journal of Physiology. After that, we will have a Q&A session. So all your questions are more than welcome. You can submit your questions within the Q&A function here on Zoom, which is located at the bottom of your screen. So please all submit your questions there. Normally, you could also upvote questions from other attendees that you would like to see answered. After that Q&A, we will enter a 30 minutes networking session where we can further discuss the paper and all things related to science. So we all hope to see you there. A link to this networking session will be posted in the chat function at the end of the session, or it's already there, I see. So you can use that link to join the session afterwards. And as a last note, today's session will be recorded and will be posted on the website of the Journal of Physiology later on. Okay, with all these things out of the way, I'll just briefly introduce why I selected this particular paper. My own research is mainly focused on the acute and chronic responses to exercise training, and more specifically on how and why exercise training is such beneficial for our cardiovascular and metabolic health. And I'm always very intrigued by every paper that shows the real potency of exercise training for preventing and countering the adverse effects of so many chronic diseases. And that's why I thought this paper is really suitable to discuss in today's Journal Club session. It shows on the importance of the capillary network and the status of the microcirculation for skeletal muscle function. And the authors, I think, really elegantly show this with two independent experimental interventions, where both interventions convincingly show how important the skeletal muscle is and how they pro-angiogenic therapies are for skeletal muscle function. But that's more than enough for me, I think. I'll now pass the word to the two lead authors of the paper. So Dr. Peter Tickle and Dr. Paul Hendrickse, take it away.
Thank you very much, Thibaux. So I'll load up my presentation here. Here we go. Okay, so jumped ahead there. So thanks for the invite again to the Journal Club today and the nice introduction. I should point out that we'd like to thank other people involved in the paper also. So Andrew Weightman was involved in designing some of the equipment we used in the exercise part of the experiment. Hakam Nazir was a PhD student at Leeds at the time who helped with the experimental work, and also to the senior authors, Professors Degens and Stuart Egginton, who conceived the idea and gained the funding to do the experimental work, from which we were able to do these publications that I'll outline today. And specifically, we're interested in the effects of chronic heart failure on skeletal muscle. And one of the important things in heart failure is that the hallmark symptom, as well as the cardiac pathology, obviously, is there is a pronounced exercise intolerance in patients. And obviously, this is very bad for them because it prevents any sort of real rehabilitation efforts, and also affect quality of life in terms of being able to walk around, do general activities day to day. So it's not entirely clear what underpins this exercise intolerance, but there's mounting evidence to suggest that it's not the cardiac pathology per se by itself that's doing this. There are peripheral factors that are affecting the exercise intolerance. And what I mean by that is there seems to be dysfunction in the skeletal muscle that occurs with disease that sort of underpins the reduction in exercise performance. And in particular, there is evidence to suggest from experimentally induced heart failure and also from clinical biopsy samples from patients that there is a microvascular rarefaction. We see a number of papers here I've put here on the left. There's a reduction in the number of functional capillaries, that is the number of capillaries there are in muscle that can carry blood and sort of deliver oxygen to the respiring tissue. And this occurs in heart failure. Okay, and so this tallies rather nicely with the paper we published last year from the same group in the Journal of Physiology, this was, and that was on the fundamental role of the microcirculation in otherwise healthy skeletal muscle tissue and how that affects exercise performance. And what we did there was to develop a surgical model whereby we could knock out capillaries within a target muscle in the hind limb and to see what the effect was on exercise performance of that muscle in the lab. So we stimulate the extensor digitorum longus under anesthesia and see how well it tolerates force decline, if you like, over a 3-minute stimulation protocol, and how that is affected by the number of underlying capillaries that are available in the muscle to blood flow. And what we see is that with a decreasing perfused capillary supply, we have a decreasing fatigue index. So what this means is that the muscle is able to perform less well with fewer capillaries available. So clearly we've shown in otherwise healthy tissue that microvascular density is an important component in describing skeletal muscle function. And we know from previous papers that in heart failure, we see a reduction in microvascular density in skeletal muscle. Well, it seems obvious, therefore, that we should go on to this to see what happens in chronic heart failure and whether the rarefaction of capillaries in there can be reversed. So in the paper we're discussing today, we had 3 clear hypotheses, and they were whether compensatory cardiac hypertrophy or the sort of incipient stage of heart failure, whether that has adverse effects on the muscle microcirculation and muscle performance, so fatigue resistance. Also, whether aerobic exercise prevents any adverse effects of cardiac hypertrophy on skeletal muscle, and also whether functional overload, which is analogous to resistance exercise, has a restorative effect on any changes in skeletal muscle in terms of its capillarization and functional output, and how that is affecting heart disease. And so to do this, we developed another surgical model. So what we wanted to do was to induce cardiac hypertrophy in a rat model. So this is a Wistar rat, all male animals. And by implantation in recovery surgery of an aortic band, this was a small titanium clip which restricted the suprarenal abdominal aorta. Okay, so it's so the head's at the top here, tail's down the bottom of the picture. And what this clip is doing is restricting blood flow to the hindquarters. And so this is causing a hypertension above the clip by way of activating the renin-angiotensin system of the kidneys to regulate blood pressure. So we're reducing flow to the kidneys. That's responding by increasing cardiac output to try and equalize the flow below the clip. So this is a long-term intervention of around 4 weeks, but I'll go through our group structure here that we used in the paper. So we had 6 distinct groups. We had a sedentary control group of rats. We had rats that were able to exercise for 4 weeks as they wished. So there's a wheel in their cage, but these were not carrying an aortic band. Similarly, non-banded overload group. And what I mean by this is that these animals were sedentary, but they underwent a surgical procedure to cause a functional overload on the EDL muscle. And we do this by removing the tibialis anterior under a relatively straightforward recovery surgery. And this causes the remaining EDL to be functionally overloaded, so it has to do more work to move the foot, and it also causes a stretch. And what we typically see in healthy tissue is an increase in mass of around 20% within 2 weeks, and an angiogenic, a powerful angiogenic response where we get an increase in capillary density. Now we had comparable groups, but also carrying an aortic band. So we had a sedentary banded group, which stayed in the cage for 4 weeks after band application. We had an exercise group which could run on a wheel while also carrying an aortic band. Now, the band was placed in these animals and then they were able to exercise for 4 weeks, at which time they were sampled. And the final group was AOB, so aortic banded for 4 weeks, after which time we imparted the overload on the EDL muscle and left that to develop for a further 2 weeks. Now, at the end of these periods, we measured under a terminal anaesthetic protocol in the lab various physiological measurements. So we could see what happened both to the cardiovascular system, but also what happened to the skeletal muscle in terms of functional performance. You can see in this graphic on the right here, this is a typical setup in the lab. So we have a very deeply anaesthetized rat hooked up to Aurora force transducers here. We measure both ipsilateral and contralateral limbs simultaneously. So the EDL muscle is connected to a force transducer, and we can stimulate the EDL muscle on both sides via an electrode. And we do this for 3 minutes to impart fatigue at 10 hertz. And this gives a decline in force output of the muscle, which is how we define our fatigue. Simultaneously, we measure vascular flow in the femoral artery on both sides of the animal. So we get an idea of gross arterial perfusion of the muscle. We measure carotid artery pressure and tail artery pressure throughout, which also enables us to measure heart rate. And via a carotid cannula, we can also inject a fluorescent mixture of dextrin, which enables us to visualize which capillaries are open to plasma flow at the end of the experiment. And as if to prove our aortic banding sort of methodology worked, we measured the hypertensive response in the chronic groups. So we see group structure along the x-axis here. We have the control group, the exercise group, the overload group, the banded group, banded with exercise and banded with overload. So these are the chronic groups. We have another group here. This is called acute banding. And this was what happened immediately after we placed the band, within an hour. So this isn't a chronic group by any means, but you can already see there is a slight increase in pressure here. But in the chronic scenario, we get a chronic hypertension, and this corresponds very well to an increase in cardiac mass of around 30% in all of the chronically banded groups. Okay, so we have a chronic hypertension causing incipient heart failure. And I'll hand over to Paul now to discuss the results and implications for therapeutic targets in the future, hopefully.
Okay, thanks, Peter. So just before I start, sadly, I'm having an issue with my webcam, so I'm not able to go on webcam for this session. I thought I'd put that out there before I get started. So following on from what Peter said, so that the rats in the exercise groups, that were one that was not subject to the banding and one that was subject to the banding. Here you can see that after 1 week of banding, the total distance ran was lower than the exercise only group. Interestingly, the inclination to run on the exercise wheel was unaffected in animals. They simply ran less distance for each exercise bout, which suggests that the tolerance to exercise is impaired with the aortic banding intervention. If you see in the top right corner here, overload-induced hypertrophy was attenuated after aortic banding when the EDL muscle mass was compared to that of the contralateral limb. While we didn't investigate the mechanism behind this, we think that this may be due to raised levels of cytokines, which may have suppressed mTOR activity, which is present in heart failure. Also, a reduction in anabolic factors have been found in rats with heart failure, both with reduced and preserved ejection fraction. In the bottom left, you can see that the femoral blood flow reduced acutely after the clip placement. and that this was restored after in chronic aortic banding, which is the AOB. So this shows on the right, bottom right, as the fatigue resistant, the fatigue index here for the acute aortic banded animals is lower, and this is in response to blood flow to the muscle, whereas in the AOB, the chronic group, there is the femoral artery blood flow was normalized, but there was still a reduction in fatigue resistance, which is shown here as a reduction in the fatigue index. So if we go to the next slide, please, Peter. Okay, so in the top left panel here, you can see what we did to determine perfused capillaries. So prior to the removal of the muscle, whilst circulation was still flowing, Hendrickse labeled dextran was injected into the carotid artery, which led to the labeling of the perfused capillaries as those that supported flow supported the flow of the Hendrickse labeled dextran, which you can see in the green there. So after the animals sacrificed and we used the muscle in histological analysis, we stained the tissue for Graphonia simplicifolia leptin, which you can see on the top two, and this for total capillaries present. And as you can see with the rings there, there's a reduction in the proportion of perfused capillaries compared to the control in the AOB group. That is the number of capillaries that support flow. So to identify the staining of a capillary, it was identified as a capillary if it was in a place where a capillary would normally be found, i.e. not within the muscle fiber, and if the intensity was greater than the surrounding area, which was completed using a thresholding function in image J. So if I go to panel 2, the top right panel, you can see that the capillary supply area was enlarged in aortic banding, so indicating that each capillary must provide for a larger area of tissue. So there's more demand. So that means the partial pressure of oxygen distribution drops. The image below shows in aortic banding the partial pressure is impaired in aortic banded groups. So you can see at the bottom here in control, and there is the level of capillarization in the control, and in the exercise and overload groups, you can see a greater proportion, greater number of perfused capillaries, whereas in the aortic banded group, there is a lower number of perfused capillaries, and this is rescued with exercise and with overload. So if you go to the next slide, please, Peter. So from this, we can say that cardiac hypertrophy is accompanied by reduced muscle fatigue resistance in spite of normal arterial blood flow. Overload improved fatigue resistance of the muscle, but the hypertrophic response to overload is blunted in animals with cardiac hypertrophy. Also, poor performance of skeletal muscle may be explained by reduced capillary supply area. And finally, targeted angiogenic intervention to restore the microcirculation may recover skeletal muscle performance in patients with incipient heart disease. So some potential treatments could be exercise-based, and in those with particularly extreme exercise intolerance, there could be passive exercise of the muscle, which has been found to increase capillary density in the work of Hilda Helliston's group. Around 10 years ago, they found that in healthy healthy participants. So that could be one particular area of further study that may be useful in this area. Yes, that's everything. Thank you very much for listening. And we would also like to thank the British Heart Foundation, Manchester Metropolitan University, Physiological Society and University of Leeds, myself being affiliated with Manchester Metropolitan University, and Peter being affiliated with the University of Leeds. Thank you very much.
Okay, thank you very much for the very interesting and clear overview of the paper. I first give the word now to Dr. Rickards, who will tell us something more about why and how this paper was accepted in the Journal of Physiology.
Thanks so much, Thibaux, and thank you for Dr. Tickle and Dr. Hendrickse's presentation there. It was great to bring everything together into a very succinct story. So I'm a reviewing editor for the Journal of Physiology, which means that I'm kind of the intermediary between the senior editor and the reviewers, and then communicate up and down that line. So my role was to really find reviewers for this paper and then work with the senior editor and the authors to go through that review process, which in this case required a couple of revisions before ultimate acceptance of this great paper. So, so in terms of some of the highlights for this paper and what led to its eventual acceptance, I think obviously the novelty is really important, obviously for the Journal of Physiology, in terms of this assessment of the microvasculature in skeletal muscle fatigue in cardiac hypertrophy. Dr. Tickle provided some background in terms of the premise and the rationale for why this study was important. And of course, that was something that was very much supported by the reviewers, and then for me and for the senior editor to move forward with acceptance of the paper. I think also you saw from the presentation that there was a very robust experimental design where there was multiple groups with different treatments, and there were also control groups, which are very important to really make an accurate assessment of the overall results that were presented today. So the controls and the methodologies that were used were appropriate, and the data were presented in a way that made sense to the reviewers, and the statistical analyses were also appropriate considering the groups that were utilized. Another aspect of this paper which led to its eventual acceptance was obviously the impact of these findings. So there were some potential therapeutic targets that were examined that could increase exercise tolerance with heart disease, and that's obviously a big issue with individuals who have these types of clinical conditions. And so there was evidence that those targets actually could be beneficial for improving exercise tolerance and hopefully reducing the symptomology that occurs because of these types of clinical conditions. The other part of this is related to that comment regarding the potential clinical implications and translational potential of this work. And that's not always a necessary requirement for the Journal of Physiology, but in this case, we could certainly see that there were some potential translational applications of this work if it was to move into larger animal models and then obviously into the human model for developing therapeutics for individuals with heart disease. So that was also a big aspect of why this paper was accepted. And then finally, I think, and this is for everyone in the audience, in terms of responsiveness of the authors to the reviewer comments. So I know in the first round of reviews, there were quite a number of items that had to be addressed by the authors, and they did a great job in respectfully and appropriately responding to those comments. And I think hopefully the authors agree that from the first submission to the final submission, there were some great improvements in the paper that as a team of reviewers and authors, I think, eventually led to a much improved paper. So I very much encourage everyone to continue to be responsive to reviewer requests, as much as reasonable, of course. So they're my main comments that I'd like to make as part of the review team for this paper. So thank you.
Okay, thank you very much, Dr. Rickards. I think it's very interesting for all of us to hear some of the other sides of the review process. Okay, with this, I would like to open the Q&A part of today's session. So as I've already mentioned, please, you can post all your questions in the Q&A function here at the bottom of your screen in Zoom for our panelists. While we wait for some of the questions to come in, I can maybe start with one or two questions. Maybe a first one. The experiments were mainly performed in the EDL muscle. Could you comment on what you would think if you would use another muscle that is more like a slow twitch muscle, like the soleus, in comparison with a more fast twitch EDL muscle? Because I think there was recently a paper published that also showed that endothelial cells in slow or fast twitch muscles are quite metabolically different. So could you comment on what you would think if there would be differences in that?
It's an interesting question. I know previous studies using a similar aortic coarctation method have looked at different muscles in the hind limb, so soleus, tibialis anterior, EDL and others, and I found a quite different response actually in terms of what happens in terms of vascular regression and various functional outputs. And so it's quite possible that we may see a different profile in the capillary regression in these muscles. Whether or not the main pathology in terms of heart disease causes a general systemic decline in all muscles at once, perhaps, but I do take your point. It could very well be that different muscle fiber types do have a different bearing on the capillary regression. I guess it's one limitation in some respects of the modeled PO2 that we presented in the talk there, in as much as we assume that there was just one fiber type. Okay, so maybe that different metabolically active fiber types are differentially affected by the sort of knockout of capillaries in the muscle, and that's probably quite a likely thing that would happen, and could be an improvement in the way you model the muscle dysfunction, if you like. So yes, basically, I think you probably would see a different result.
Interestingly, you mentioned about the two different muscle fiber muscle types, fast and slow muscle. So within this, within the model that we use, we looked at 3 regions of interest across the muscle. So there are areas within the muscle that are faster and slower in the fiber type composition, and we found that there wasn't really a difference across the 3. But as Peter said, it could be that there is a difference between different muscles.
Yeah, I agree. Yeah, I think it would be really interesting to see if there would be any differences. I don't think there's a question in Q&A yet. So a second question from me, maybe. So we see, or you see in your paper, that muscle performance was declined. So it is possibly determined by the microcirculation in this model. I know for more human performance that, for example, for VO2 max, etc., that the central factors, blood volume, is way more important to determine that. Do you know if there are differences between rats and mice or humans in respect to importance or role of microcirculation compared to more central factors for exercise performance?
Yeah, it's a really good question, Thibaux. It's a tricky one. Yeah. So I think it all depends. Of course, we've found a fundamental link between the microcirculation, the muscle, and exercise performance. And I think the question here is what problems you get in disease determine your readout in terms of muscle performance. So with more advanced disease in, say, humans, that may be accompanied by problems in arterial and endothelial dysfunction, which then will potentially take over as a predominant cause of exercise intolerance, I think. Whether or not that is affected here, I doubt. These are pretty healthy animals by the time we place the band. So they're adolescent animals without comorbidities. And so the good thing about our model here is that we're looking in isolation of other factors that may be going wrong, basically. So if we allowed the hypertension to progress for a few months, say, perhaps we'd get further physiological problems affecting the systemic circulation as well as the local problem. And again, that would be a good model for sort of human advanced chronic heart failure. But I'm not sure if that answers your question.
If I just add to that, Peter, so there was, this paper is building on the findings of the previous paper that we did together, where we found the link between, we found a link between fatigue resistance and functional capillarization of the muscle. So we have found that there is a link between those two things. So the lower the perfusion of the muscle, the poorer the fatigue resistance of the muscle. So this was leading on from that, looking at it in a specific disease model. So we already know that it does, the perfusion of the muscle is important for fatigue resistance. This is just within this specific model where this is one of the symptoms, so to speak.
Okay, yeah, thank you very much. Dr. Rickards also has a question, so feel free to ask.
Thanks, Thibaux. So I have a question regarding the subjects that we use. So you mentioned that you had young males in this study. Do you have any comments about what may have happened if females were included, and also if you had an older age group? So obviously heart disease is more predominant in an older population. So just comments on what you might have seen if you'd included those two groups.
Yeah, that's another great question. So we'd like to work with older animals because, of course, this is where you do find these sort of metabolic diseases and so on. But the problem with using older animals is they're fantastically expensive compared to using young animals. And so that limits what we can do in some respects in the lab. So going forward, obviously, you'd like to quantify the sort of comorbidities, as I mentioned, that go along with the chronic heart failure, and age-related changes that you see in muscle may also compound these changes you see in disease. As animals get older, of course, you lose muscle mass, you lose functional output of the muscle, and you also see a microvascular rarefaction as you get older too. And so in some respects, older animals would be expected to be less able to respond, I guess, than the younger animals. So the regenerative capacity may be impaired. And so that's something we really need to test with a very well-funded project, obviously. Question about female animals, of course, again, it's a question of time and budgetary restraints also, really. So because the foundational data, the empirical data we've got already on these animals was collected in male animals, as has been the sort of fashion for a long time, we wanted to build on that using the same subset of animals, basically. But there is, of course, an obvious idea to use female animals as well. I think there's some evidence that heart disease is protected against somewhat by various hormonal effects in females relative to males. And so you may see a more pronounced or earlier case of the cardiac dysfunction occurring in males with consequent implications for skeletal muscle. But of course, we have to do the experiments, so it would be great to see that in future.
I'd also think that, so based on looking at the hypertrophy side on it, of the study, you would likely see a lower hypertrophic response in the older animals. So there's some evidence that capillary density can decrease with age, and capillarization of muscle can decrease with age, which has been shown recently to be a factor in the hypertrophic response by the work of Tim Schneider's group and Fry's group as well. So that could possibly affect the hypertrophic response within older animals.
I guess the interaction of age and sex is also important, because while younger females may have protection, once they get past menopause, then that protection may actually be diminished. So it'd be really interesting to kind of examine those questions, and certainly appreciate the issues of funding and resources to do that work. But certainly, hopefully that's on the list of things to do down the road. So thanks for your responses.
Okay, there are now some questions in the Q&A. So first question is, with muscle fatigue resistance decrease in the aortic banded animals, running on the wheel distance was slower. Curious if there was also a change in duration of each bout of running, as this will be a factor in translating the results to a human setting with willpower to perform the exercises to preventive measure.
Yeah, I think, just to clarify, I don't think the muscle fatigue in the animals that were exercised carrying a band, the fatigue resistance was normalized to intact control levels, despite the fact that they were exercising at a lower sort of intensity. But there was a change in duration, so they were not able to run as long and they were not able to run as fast, but they were able to do as many bouts, if you like. So they had the inclination to exercise as much as the healthy rats, if you like, but they were unable to sustain exercise. And so that was a very low threshold, but this was sufficient to normalize their muscle performance and to some extent their microvascular density.
So even with this reduced volume, they still had the positive effects of the exercise.
Exactly right, yeah. So that's a promising sort of finding of our research here. So even with low intensity exercise, which would suit people with heart failure or other diseases, to which you also see vessel rarefaction in, it would appear from our data that even low bouts of exercise will be beneficial.
That's indeed very promising and good to hear. A second question, the lack of muscle growth in response to the overload model, did you collect blood samples, which you could use to assess inflammation markers as well as stress hormone concentrations, as both are known to drive muscle breakdown?
Do you want to take that one, Paul? I think that's your problem.
Yeah. So we did attempt to do this. We had a lot of issues with the kits that we were using, and as you can imagine, you only have so much of the sample available, and so much funding for the to buy the kits that we would have used. So there wasn't really, it didn't really pan out as we had planned. It would have been nice to include that, but it wasn't integral to the main points of the paper and the hypotheses of the paper.
Just add to that, I think we were anticipating, given what we saw in the muscle growth or lack of in the aortic banded model, we were expecting a sort of inflammatory effect, or expecting to see that in the molecular analyses that Paul did. But I got the feeling we didn't really get a definite answer, and this sort of corresponds with previous work in humans, where you get a transient, very high release of inflammatory markers after a cardiac event, which then sort of decrease over the recovery period. And because we were sampling 4 weeks after placing the band at a minimum, 6 weeks for the overload, it's quite possible that these levels have gone down to very low anyway, and the damage had been done, if you like. So without a sort of longitudinal study where we sample at a number of time points, it's difficult to tell exactly what was happening. So again, that's a great idea for the future. We should be taking blood samples as we go along, as the disease develops.
Yeah, I agree. It would definitely be interesting to see the temporal relationship as well. The next question here in the Q&A. Any idea on the mechanisms involved in the decrease in capillary density?
Yeah, it's not entirely clear so we didn't really see a decrease in the anatomical supply of capillaries. So all the capillaries appeared to still be there. It was the functional capillaries that were decreasing. So of those anatomical capillaries, some of them weren't perfused anymore. So because we have a hypertension, systemic hypertension, that's associated with capillary closure, so vasoconstriction. So it could well be that some of these capillaries were just blocked by way of smooth muscle activation. And when with exercise provision, that could have, via mediators such as nitric oxide release, caused these capillaries to reopen. And that's why we see an apparent increase in functional capillary density. Otherwise, it may just be a cause of them being closed for a while due to this vasoconstriction actually breaking down. So if we left the samples to run a bit longer, the animals to live a bit longer, we may have seen an anatomical decrease as well as the functional decrease. So we could be in the early stages here of a capillary turnover situation.
Okay, and Paul, did you have anything to say?
Sorry, my mistake. I pressed the wrong button then.
No problem, no problem. Another question that I had, so you see this capillary rarefaction and these problems in the microcirculation. Would you think this would also hamper the metabolic health of the animals? Because the microcirculation is, of course, also really important for insulin sensitivity and other metabolic markers.
I think that's right, yeah. So because if you get vessel rarefaction at a grand scale, you're sort of basically in diabetes territory, aren't you? Yeah. So yeah, longer term, I think you'd definitely expect that. And that sort of links with Caroline's point with older animals. You'd expect, these sort of metabolic disorders to come alongside the other disorders. So chronic heart failure may be accompanied by diabetes or a kidney failure as well. And so all these things tend to link up and cause dysfunction at the level of the muscle in terms of the vascular, the mitochondrial, the muscle fibers, and so on.
Mm-hmm. Okay, I think we have time for one or two more questions. So if there's any question that you would like to ask, please put it in the question Q&A. There it is. Did you verify capillary rarefaction in other organs?
I'm afraid not, no. It's a simple answer.
And what would you think? Would there also be rarefaction in the other organs?
I suspect so, because we're looking at a cardiac pathology, basically. We're looking at hypertrophy here. Presumably, it is a systemically derived problem from the hypertension above the band. And so I would expect other muscles to be similarly affected and potentially other organs. Obviously, we haven't tried that, but potentially, yeah, sure. Maybe the cardiac muscle also was affected.
Mm-hmm. Okay, so maybe one final question. With the chronic, in the acute banding, you saw a decrement in the femoral blood flow, while with the chronic banding, the flow wasn't restricted anymore. Could you comment on possible mechanisms for that? Yeah,
so we measured the femoral artery flow as the band was being placed, and that accompanied. So as soon as you put the band on, you get an immediate drop in flow. And this is before the homeostatic mechanisms have sort of ramped up to increase the pressure above the band to normalize the pressure below the band, if you like, so we can regain flow. So I think it takes a few days for those mechanisms to take over to enable the cranial side of the band to increase the pressure, to get the tail pressure or the hindquarter blood pressure and blood flow back to where it needs to be. Perhaps this isn't the case in older animals, again, where you get these sort of endothelial dysfunctions creeping in. But yeah, I think it was a transient phase, which says more about the rats being able to normalize it than a disease per se.
Yeah. Okay, one final short question here in the chat. Did you find any anomalies between capillary density and TPR? So I think peripheral resistance, they might.
Yeah, I'm not sure what TPR stands for. Are you, Peter?
No, not entirely.
Total peripheral resistance or something?
Well, we're assuming that was relatively unaffected because the femoral artery flow was as normal in the chronically banded animals. So in effect, it was normalized, irregardless of the change in capillary density. That's not to say, I mean, the capillaries, the flow through the capillaries could have changed, you know, the transit times could have changed. A lot of parameters can also vary at the level of the microcirculation as well as the arterial circulation. So, you know, it's not a question of one or the other. It's more than likely a sort of blend of these things.
Yeah, as with a lot of things, it's probably a combination of several mechanisms going together. Yeah,
that's it.
Okay, I think all the questions in the Q&A are answered at least. Okay, with this, I would like to end this part of the Q&A session. Thank you for all the attendees for posting your questions, and of course, thank you to our panel for being here today, for presenting the paper, for answering the questions. I would just like to remind everyone that there's a networking session just after this, a 30-minute networking session. The link to join us will be posted in the chat. It was posted in the beginning. It is now posted again, so everybody can click the link to join it. And with this, I would like to end, and I hope to see you all in a couple of minutes in the networking session. So thank you all.